So this is chapter 4. So in chapter 4, we're going to learn about the anatomy of prokaryotes. So the first thing we want to do is compare and contrast prokaryotes and eukaryotes. So in prokaryotes, the genetic material or the DNA, it's just, a circular single chromosome typically. So it's only one chromosome. Most of the time, that's all it is. There are some exceptions where it's found. They're actually found, what did I do? Somehow I moved they're found in this region in the cell called the nucleoid. Okay, so prokaryote means that it's before the nucleus. So there's no nucleus in prokaryotes. So you need to remember that. Okay, eukaryotes, the genetic material is typically linear. Let's go back to one. Linear chromosomes. And you guys know where they're found. They're found in the nucleus. Okay, so I'm just trying to beat the point home that prokaryotes, presence of a nucleus. No nucleus. Okay, but eukaryotes have one. So definitely. Okay, now, prokaryotes have no membrane-bound organelles. Okay, so nothing with the membrane around it. So no membrane-bound organelles. So nothing with a membrane around it. So they don't have a nucleolus. Okay, they don't have an endoplasmic reticulum. They don't have chloroplasts, and they don't have a Golgi apparatus. But they do have ribosomes. Okay, because if you remember, ribosomes are just protein and RNA. There's no plasma membrane around them. So they do have ribosomes. Okay, now eukaryotes, they have all of those. So yes, they have all of those organelles. Okay, now enzymes for respiration. So the enzymes for cellular respiration for prokaryotes are found in the plasma membrane. Okay, so they're found in the membrane. We're going to talk about this a little more. Okay, but in eukaryotes, where are they found? Okay, so they're found in mitochondria. So would a prokaryote have mitochondria? No, right? Because there's no membrane-bound organelles. So no mitochondria in prokaryotes. Okay, now prokaryotes, I'm trying to move this. It's not letting me, I don't know how I moved it accidentally. Okay, so extra chromosomal DNA. So bacteria are different. They do have some DNA that's not that's not a chromosome. They're actually called plasmids, and we'll talk about them more in a minute. Now, eukaryotes typically don't have them. So they don't have them unless we put them in there, like a human adds it. Sorry. Okay, now, cell walls. So prokaryotes do have cell walls, and that's what makes them distinct. Okay, most eukaryotes do have a cell wall, except for mammal cells. Right, and we don't have them. Okay, flagella, yes. Cilia, yes. Flagella, yes. Cilia, yes. Okay, bacteria use cilia to move. Okay, and then pili. A pili I'll explain it more in a minute, but it's an extension off of the bacteria. So it just kind of goes like this around it. I have a whole slide about it. So bacteria have them, but eukaryotes don't have them. I don't know what I did. Okay, now how do they reproduce? So remember learning how eukaryotes reproduce, right? They have to go through mitosis and meiosis, right? To go through reproduction. They can be sexual reproduction, but some things have asexual reproduction. So if you think about maybe a plant, when you're trying to grow, you take a leaf or a stem, right, and you like clone it. That's an example of asexual reproduction in eukaryotes. Now, in prokaryotes, cell division, it's really, really simple. It's just called binary fission. It's so much easier. Okay, it's really, really simple. And reproduction is going to be asexual. Okay, so there's no male or female bacteria. They're just all kind of the same. They're all neutral. Okay, now a typical prokaryotic cell. So here are the things that we have, and we've kind of maybe hinted on some of them. Okay, so here is here, let me try to point. Let's see if I can find a pointer. No, hold on. There's a pointer. I think. Yeah, there's a pointer. So here's the chromosome. Okay, and it's in this area called the nucleoid. So it's just kind of in a region. It's actually attached to the cell wall. It's not floating around in there freely. So it's attached to the plasma membrane, I should say. So it's attached in there. Okay, and then here's a plasmid. So a plasmid's just a circular piece of DNA. It's extra chromosomal. So let's write that on here. So plasmid, it's an extra chromosomal piece of DNA. Okay, it can reproduce by itself, and it can replicate and make proteins. Okay, now the cytoplasm is the same, right? So there's the cytoplasm. It's basically the same thing. There's ribosomes, and the ribosomes can be kind of lined up and stuck together in little chains. They call it polyribosome. We'll come back to inclusion. Flagellum, we're okay with. We've seen that before. Pilus are the parts that stick off. So in this case, these little things that are sticking out, those are fimbriae. Now, in here, if we start on the inside, we're going to have the cell membrane, which is just like a plasma membrane. Then we'll have a cell wall. Okay, and then we'll have this outer layer, which is the capsule or slime layer. Sometimes it's called a glycocalyx. So we're going to go over everything that's in here, and we'll also talk about inclusions. Okay, so why do cells have cell walls? So the main reason that these bacteria have cell walls, what they do is they're going to maintain the shape of the cell. So they're going to keep it as a sphere, they're going to keep it as a rod. Okay, now, the cell wall can have holes in it, okay, but the holes or the pores in it don't have anything to do with cell transport. They'll help protect the cell from blowing up if too much water gets in, but they don't have protein pumps, or there's no channels or anything like that. So we don't have to worry about cell transport with the cell wall. Okay, now the main component that makes up a cell wall in bacteria is called peptidoglycan. So if you guys look at that word, right, you could figure out some things about it. Oops, wait a minute. Okay, so peptido, right, would be peptide or proteins, and glycan would be sugars. So peptidoglycan is a polysaccharide. Okay, so it's got this sugar backbone kind of going like this. So these are the polysaccharides. Okay, and then they are connected together by amino acids, by peptides. So it kind of makes like a chain or a wall. And they know exactly what sugars are here, and they know exactly what amino acids are here. But you don't have to memorize those. I just want you to know that that's what it's made up of, and it's kind of like a bridge. There's a picture in your book on page 83 about it. Okay, now we're going to classify things as gram-positive or gram-negative, right? So gram-positive cell walls, those are going to be the ones that look purple, The gram-positive cell walls have a lot of peptidoglycan in them. So 90% of the cell wall is made out of peptidoglycan. So it's really, really thick. So, well, really, 30 to 60 nanometers, so not that thick, but thick enough. Okay, so here it is. It's this purple layer, and the book makes it purple so you can see it. Now, in them, there's this thing called teichoic acid. So the teichoic acid are these red things that are sticking up. Okay, the teichoic acid are places where bacteriophages, which these are viruses, we'll talk about them more later, they can attach. So it's almost like a receptor. They're negatively charged, the teichoic acid is. Sometimes cations can bind here and they can go into the cell that way. But the big thing is this a whole big thick layer of peptidoglycan. Okay, so here's the cell wall, and there's a little bit of space. Okay, and then this is the plasma membrane. I hope that makes sense. Okay, now gram-negative cell walls, they're completely different. Okay, so they have two layers of membrane. So they have an outer layer here, and then they have an inner layer. Okay, the outer layer has these things called porins in it, which are pores, and then they have some receptors in them. But they also have this thing called lipopolysaccharide, and I have that on the next slide. So gram-negatives have lipopolysaccharide, which is going to be really important. Okay, the peptidoglycan layer in gram-negatives is really, really thin. So it's only 10 to 20% compared to 90% of the wall. So it's this thin purple layer here. Okay, and then, let's see, it's this thin purple layer, like right in here. Okay, and then this is the plasma membrane. Try to get my pen situated. I can't find the black. The black ink is gone. I don't know what I did. Hold on. I don't know. So this is the plasma membrane. So it's different, right? So it looks different. So this one they're making it so it looks red. Okay, so there's a little bit of space between this peptidoglycan and the plasma membrane, and they call that periplasmic space. Usually you only see it if the cells are dead. Okay, and then plasma membrane. Okay, now lipopolysaccharides, they're also called LPS, or sometimes they're called endotoxins. So these are only found in gram-negative bacteria, and here's what they look like. Okay, so it's some sugars, right? So this polysaccharide, and then coming off of the polysaccharide, there are these fatty acid tails. So it's actually a toxin, so right, endotoxin, so it's toxic. So the part that's toxic is this lipid A part, the bottom part, the part that's hydrophobic. Okay, so what it does is it's a pyrogen, so it causes a fever. So pyro is like pyromaniac, so fire. So it heats your body up. It causes your blood vessels to dilate. Okay, and when your blood vessels dilate and get big, your blood pressure will go down, and that'll result in shock, right? So that's not good if your blood pressure crashes. So the only time lipopolysaccharide is released is if gram-negative cells are broken apart. So if the cells are lysed or dies, they break apart, and then this is released, and it can cause a problem. Okay, so it could cause a problem if somebody has a gram-negative infection. It could also cause a problem because, so E. coli is gram-negative, and it's used to make different things. It's used to make drugs, so biomanufacturing, and you don't want any of the lipopolysaccharides in there. So we'll talk about that more as time goes on. Okay, so here's another picture I got from a different place to try to show you gram-positive versus gram-negative. So gram-positive, see this is all the screw up from when I imported it. It looks weird. Okay, so gram-positive cell wall. Here's all the peptidoglycan, so the layers. Okay, and what happens is you add the purple, right, and you add the grams iodine, and then in the alcohol step, what happens is the pores close, so the little holes in there close, and the crystal violet and the iodine can't leave, so this stays purple. Okay, in the gram-negative, what happens is when you go to wash it with the alcohol actually dissolves this layer. Okay, so it dissolves the outer membrane, and the crystal violet and the iodine leave. Okay, and then it ends up looking with no color. Okay, so you lose everything until you add saffron in, and then they'll look red. So this is the basic idea. So I want you to get big peptidoglycan layer, right, little space, and then the plasma membrane. Okay, then you have a membrane, a little peptidoglycan layer, and then another membrane in gram-negative. Okay, so now in bacteria, we can actually remove the cell wall and not kill it if you're careful. Okay, so if you need to do some work with bacteria, you can use enzymes and they'll digest the cell wall. And if you can stop that reaction, you can end up with this thing called a protoplast. Okay, and a protoplast it's got all the information of the cell, it just doesn't have the cell wall. So you can kind of mess with this genetically, and then you can, if you baby it and take care of it, then you can bring it back and go back to here. So it's interesting. Now, in order to control bacteria, there are methods that actually destroy the cell wall. So penicillin, it blocks the formation of the peptidoglycan layer. So if you can't form the peptidoglycan layer, you don't have the cell wall, and the cell will lyse. Lysozyme, digest cell walls, and so that's found in our tears and in our saliva. So that helps protect us from bacteria. Okay, now there are some other types of cell walls besides the gram-positive and gram-negative. So there's the acid-fast cell wall, and that's found in mycobacteria. They have less peptidoglycan and more lipids, so the lipids make them hydrophobic or are pretty waxy. So I mentioned this before, so the organism that causes tuberculosis and the organism this causes leprosy. So mycobacterium have this kind of wall. So just know that it's lipidy. So we have to use an acid-fast stain. You can't gram stain. Okay, now there are some organisms that are considered bacteria, but they don't have a cell wall. An example of those is mycoplasma. So mycoplasma are pleomorphic, so the shape changes because there's no cell wall. So they're like all sorts of different, kind of sizes of rods. So they grow really, really, really slow. And if you do cell culture in a lab, they can be found as contaminants, so they can cause some problems in research labs. There are some bacteria that, for some reason, they've lost the ability to form a cell wall. So there aren't that many of them, but if they lose it, they call them L forms. They found these types of bacteria associated with some chronic or recurrent diseases. So there's one that they found with Crohn's disease. So they're not quite sure. There's a lot of research. They're still trying to figure this out. So they don't know exactly what's going on, but they do exist. Okay, so basically the prokaryotic cell membrane is the same as any other membrane that you've looked at. So cell membrane, plasma membrane, same thing. So it's phospholipid bilayer. There are proteins in it. It follows the fluid mosaic model. So I think you guys are familiar with that from either anatomy or biology. So hydrophobic, hydrophilic, phospholipid bilayer. Okay, now we'll add some more things. This bugs me because things get messed up when I import it. I have it formatted so nice. Oh well. Okay, so internal structures. So We have cytoplasm in bacteria, so it's just a semi-fluid substance that's inside. Now, ribosomes, you remember that's the site of protein synthesis. So ribosomes are made of protein and RNA. They have a large and small subunit. There is a difference between eukaryotic ribosomes and bacterial ribosomes. So when we're trying to figure out how big a ribosome is, or how we describe them, is described by this concept called sedimentation rate. So sedimentation rate is kind of based on size and density. They use this unit called S, and it's some guy's name, and they just use S as abbreviation. What you kind of need to understand is bacterial ribosomes are smaller than eukaryotic ribosomes. So they are different. It's a different sequence of amino acids. So they're distinct. So it turns out that some antibiotics, like streptomycin and erythromycin, can bind to this 70S ribosome, and they interfere with bacterial protein synthesis, but they don't interfere with human protein synthesis. So it's kind of a slick mechanism. Okay, now the nuclear region is called the nucleoid. So typically there's one circular chromosome, and it is attached to the plasma membrane. It's not just randomly floating in there. It's kind of like tacked on. There are some exceptions to this one thing. There are some things that have two, but their chromosomes aren't the same size. And so there are some variations to this one chromosome. Now, plasmids are interesting. Okay, so they naturally occur in bacteria. Okay, so they naturally occur in bacteria. It's just a circular piece of DNA. They replicate independently of the chromosome. Okay, and we can put genes in them. We actually manipulate them now. Scientists can manipulate them. So a scientist could take a plasmid, they could insert a piece of DNA into it, and they could get this plasmid to make a protein that matches that piece of DNA. So we'll talk more about this in the little biotech chapter, but plasmids are different. Okay, you haven't seen them in eukaryotes. Plasmids do carry the genes for antibiotic resistance. Okay, so some of the antibiotic resistance that we have is coming from these plasmids that naturally occur in bacteria. Okay, so more things that are in here. Chromatophores. So chroma is color. Okay, so they're found in photosynthetic bacteria or bacteria. These are called cyanobacteria. So it's pigments. The pigments are in there. This picture shows it, the green, right? It's real pretty. The green captures the light. So like and you can do photosynthesis. Okay, and there's other variations, but this is one thing that you can have the pigment, like the chlorophyll in there, that you can use to capture light and make energy, make sugar with. Okay, now inclusions are broken into two categories. So there are granules and there are vesicles. So granules are not membrane bound. So like in our muscle cells, we have inclusions that are glycogen. So they're clumps of a molecule that are found in the cell. So bacteria have them too. So here's a clump of carbohydrate. Well, it's not like really easy to see, but there can be inclusions or clumps of carbohydrates in the cells. Sometimes bacterial cells have vesicles that have gas in them, so they look like they're empty. Sometimes they contain other chemicals like iron, or some of them, there's some that they're like magnetic. So, okay, now endospores. Endospores are not for reproduction, okay, even though sometimes people just say they're spores. Their endospores aren't for reproduction, so it's not like a seed, but what it is it's a resting structure. So if environmental conditions get bad, so that the bacteria start to run out of food or water, what they'll do is within the cell, they'll form this spore, this resting spore. Okay, so there's a core, there's a middle part, okay, there's a cortex that's around it, and then there's this spore coat. Okay, so in here is enough stuff that if the conditions become good again, this can start to grow and become bacteria again, or be a bacteria, kind of change and be a cell. Okay, so there's probably 20 genera that have spores, but 2 have been studied a lot and are really important, and so you need to know these 2, Bacillus and Clostridium. So they're both gram-positive rods. So you need to know Bacillus and Clostridium, and we'll come back and talk about them more. But they form spores. So the spores can be in the soil, and then the soil, they can just be in the soil. Like some of them have been around for like 10,000 years. It's crazy. So they find samples of maybe Arctic ice, and they go in and they find spores in it, and they can put the spores on media and get the spores to grow. So it's kind of crazy. So they last for a really, really long time. The other thing is they are really hard to kill if they're someplace where you don't want them to be. So it's hard to autoclave them. It's hard to bleach them. So they contain this dipicolonic acid, and they also contain lots of calcium. And for some reason, that helps them survive. They don't have much water in them, so that's helpful too. One organism, so Bacillus anthracis. Okay, so that's anthrax. So bioterrorists take advantage of the fact that this can make spores to weaponize it. So you grow it up, you treat it bad, right? Take away its food, and then it forms spores, and the spores survive, and you can mail them out and cause disease. Okay, so stuff that's on the outside of the cells, so external structures. So 50% of bacteria can move, so we have to talk about flagella. So if they don't have any flagella, they're atrichous, so that means they don't move, right? So trichous has to do with hair. All cocci don't move, okay? So if you have a spherical organism, it shouldn't move, okay? There are bacilli, there are rods that don't move. Okay, so monotrichous means they have one at the end. Amphi means they have them at both ends. Lopho means they have more than one on each end. Okay, and then peri means they're covered in it, so that's peri. Okay, now, another type of movement is generated by this thing called axial filament. So spirochetes have a protein that goes all the way down it. I always kind of think of it like inchwormy is how it moves. So they don't move with flagella, but they move with this axial filament. So only spirochetes have that. Okay, now, how do bacteria move? So when bacteria move, okay, it's pretty random, usually. Like they're just kind of moving around and somehow they end up someplace. Okay, when they go to run, they call it run when they move in a straight line. So all of the flagella, okay, kind of get their act together and they push it forward in a direction, and that's called running. Okay, then the flagella will lose it and they don't have their act together, right? And the bacteria are just tumbling around. So they'll tumble and then they'll run. And then they'll tumble and then they'll run. And most of the time it's usually random. Now, they've found that bacteria do exhibit these two things called chemotaxis or phototaxis. So positive chemotaxis means that a organism, bacteria, would actually move towards a chemical. So this red one, it's moving towards something. Okay, negative chemotaxis means it's repelled, so it's moving away from a chemical. So in a way, this could be a way to maybe protect something. Like if you could coat a catheter with something that's a repellent, the organisms wouldn't want to go towards it. Okay, bacteria also exhibit phototaxis. So phototaxis is they can be attracted or repelled by light. Okay, now more external structures. So pilus is singular, pili is plural. So the general function of them is for attachment, and they're made out of a protein that's called pilin. Here there's two bacteria that have fimbriae. So the fimbriae are attachment pili. So they have these extensions. So it's almost like cytoplasmic extension coming off of them. And it makes some kind of like Velcro. Okay, so the fimbriae make them attached to things. So they could, if they're growing in a tube and you get a layer right here that's called a pellicle, lots of times they have fimbriae, and the fimbriae is going to help keep them at that layer. Sometimes these fimbriae allow them to attach to body parts. So there are some STD causing agents that have these attachment pili, and those will attach to genitals or even to sperm and be transmitted. So if you have these, lots of times those organisms are more pathogenic. Okay, now another type of pili is called a conjugation pili. Sometimes it's called a sex pili or F pili. And what they do is they form a bridge or an attachment between two organisms. Then maybe a plasmid can be transferred from one organism to another. So it's bacteria sex. That's as good as we get. Okay, now the last thing is the glycocalyx. Okay, so two types or two ways to describe the glycocalyx. So the glycocalyx, here's the cell, right? And then on this outside of the cell, okay, so it's got carbohydrates in it, got proteins in it. It's this outer layer. Okay, so if it's really thick, they call it a capsule. Okay, so it usually consists of a complex polysaccharide. Okay, so it's a carbohydrate that's in a gel. And each strain of bacteria has a different polysaccharide. It helps protect this organism from the immune system. So the immune system can attack the organism as well if it's got a capsule on it. And the capsule can also make it sticky. So typically, if something has a capsule, it's going to be more pathogenic. Okay, now another way that they refer to this is also a slime layer. So a slime layer would be a thinner glycocalyx, and it's usually bound to the cell wall. It helps protect the cell against drying, and sometimes it'll bind nutrients, so it gives a little bit of an advantage. Now it can stick to things too. So it could stick to a catheter. It could stick to, I always think of your dog water bowl. So your dog water bowl, if it gets slimy, the bacteria are settling on the bottom and they're starting to form a layer. Okay, your teeth in the morning before you brush, you know how they feel filmy maybe? Okay, that's a biofilm. So biofilm was just layers with these slime layers stuck together. They can be a medical concern because they can, biofilms can accumulate in catheters or like airway tubes, things like that.